हेलो एवरीवन एंड वेलकम बैक टू द वर्ल्ड ऑफ बायोटेक्नोलॉजी। मेरी जो आज की वीडियो है दैट विल बी ऑल अबाउट ऑन इफेक्ट ऑफ डिटर्जेंट ऑन आरबीसीज। इसमें हम बात करेंगे कि अगर मैंने आरबीसी ली है, अब समझिए कि हमारा जो ब्लड होता है, अगर मैं बताऊँ आपको तो ब्लड के अंदर हम बात करते हैं कि वो दो चीज़ों से बना है, प्लाज्मा और ब्लड कॉर्पिसल्स। ब्लड कॉर्पिसल्स के अंदर हम बात करते हैं आरबीसीज, डब्ल्यूबीसीज एंड प्लेटलेट्स। आरबीसीज को हम लोग एरिथ्रोसाइट भी कहते हैं, और जो हमारी आरबीसीज होती हैं वो बाइकॉनकेव इन शेप होती हैं और वो रेड कलर की होती है, और बेसिकली वही रिस्पॉन्सिबल होती हैं जो कि हमारे ब्लड को रेड कलर देती हैं। तो अब अगर हम इन आरबीसीज को डिटर्जेंट में डालेंगे तो उस पर क्या इफेक्ट आएगा वो हमने पढ़ना है। सो लेट्स गेट इनटू द वीडियो।
The requirements of the experiment are blood sample, syringe, Eppendorf tubes, slides, microscope, cover slips, weighing balance, beakers, 0.1% sodium chloride, 0.9% sodium chloride, 1% sodium dodecyl sulfate, 1% cetyl trimethyl ammonium bromide, and distilled water. This experiment is based on the concept of osmosis. Osmosis, as we all know, is a process which involves the movement of water molecules from a region of low concentration to a region of high concentration. For easy understanding of osmosis, we place RBCs in different solutions, that is in isotonic solution, in hypertonic solution, and in hypotonic solution. In isotonic solution, there will be no change in the shape of RBCs, whereas in hypertonic solution, shrinkage of RBCs will occur, and in case of hypotonic solution, swelling of RBCs will occur. Here in this slide, it is quite visible what are the differences when the RBC is placed in different solutions. In case of isotonic solution, it is very much clear that the RBCs have developed no difference or no change in their shape. They are biconcave and they have a disk in between. Whereas in case of hypertonic solution, shrinkage has occurred, and this had led to development of an irregular shape of RBCs. Whereas in case of hypertonic solution, the RBCs have undergone swelling. The procedure for this experiment is as follows. First of all, we are going to collect 1 ml of venous blood sample. Then this blood is to be mixed with different solutions, that is 0.1% sodium chloride, 0.9% sodium chloride, 1% sodium dodecyl sulfate, and 1% cetyl trimethyl ammonium bromide. Then these samples are to be incubated at room temperature for 15 minutes. After incubation, these samples are then used for making smears, and these smears should be thin. Then these smears or slides are observed under the microscope, and the observations are observed or recorded. The results of the following experiment are: first of all, when the RBCs were placed in 0.1% sodium chloride, which is an hypotonic solution, the RBCs will undergo swelling as the water from outside of the cell has entered to inside of the cell and had lead to the swelling of the RBCs due to concentration gradient or difference in the concentration. Second, when 0.9% sodium chloride is used and RBCs is placed in them, then no effect is observed as the concentration is same in interior of the cell and the exterior of the cell. Third, when the RBCs were placed in 1% sodium dodecyl sulfate and 1% cetyl trimethyl ammonium bromide, the concentration gradient is there. So the water molecules will move from the cell to the outside of the cell, and this will lead to shrinkage of the cell and development of an irregular shape. This is due to the concentration gradient. This slide clearly shows that when these slides are observed under the microscope, these are the observations. First diagram clearly shows the structure of the RBCs, that is, they are normal. Whereas in case of hypertonic solution, change in the shape can be easily observed as they have undergone shrinkage. Whereas in hypotonic solution, they have undergone swelling. So whenever you're performing this experiment, following things need to be kept in mind. First of all, the blood which is to be collected should be handled properly with wearing or while wearing gloves only. in order to avoid contamination. Second, the solution which is to be used as a detergent for determining the effects of detergent or RBCs should be prepared very much carefully and with accuracy. And third, the slide which are to be used for making the smear should be sterilized and clean before use, and the smear which is to be made from the sample should be thin. Thank you.